Purkinje cells, named after John Evangelista Purkinje, stand out as a significant type of sizable neuron present in the cerebellar cortex. These cells play a key role in regulating motor functions. Notably, Purkinje cells release the neurotransmitter GABA, which helps to inhibit certain neurons, thereby reducing nerve impulse transmission and effectively controlling the body's motor activities. Structurally, Purkinje cells, among the largest neurons in the human brain, boast elaborate dendritic arbors with numerous spines. Found in the cerebellar Purkinje layer, these cells align akin to stacked dominoes, forming two-dimensional layers with parallel fibers passing through their dendrites. While parallel fibers make weaker excitatory synapses, climbing fibers deliver potent excitatory input. Each Purkinje cell interacts with around 500 climbing fiber synapses, establishing a complex computational system within the cell. Moreover, both basket and stellate cells provide GABAergic input to Purkinje cells, further enhancing their functionality by synapsing onto various parts of the cell. Within the molecular realm, the Purkinje layer expresses a multitude of unique genes, playing pivotal roles in cellular function and development. Noteworthy examples include Purkinje cell protein 4, PCP4, vital for calcium regulation in Purkinje cells, impacting synaptic plasticity and locomotor learning. Regarding development, research on mammalian embryos has elucidated the origins of Purkinje cells. These cells arise from progenitors in the ventricular neuroepithelium, migrating to the cerebellar cortex's outer surface to form the Purkinje layer. During cerebellar neurogenesis, specific transcription factors like neurogenin 2 and neurogenin can play crucial roles in specifying Purkinje cell subsets, contributing to the intricate cerebellar topography. Furthermore, evidence suggests the potential involvement of bone marrow cells in generating or integrating with Purkinje cells, hinting at their role in central nervous system repair. Additionally, a common stem cell ancestor may exist among Purkinje neurons, B lymphocytes, and certain cells in the human adrenal cortex, pointing towards a shared developmental origin. Purkinje cells exhibit two primary types of electrophysiological activity. Simple spikes can occur at various rates, ranging from 17 to 150 Hz, either spontaneously or upon synaptic activation by the parallel fibers originating from granule cells. In contrast, complex spikes manifest as slow 1/3 Hz spikes, featuring an initial prolonged large amplitude spike, followed by a burst of smaller amplitude action potentials at high frequency. These complex spikes arise due to climbing fiber activation, potentially leading to the generation of calcium-mediated action potentials in the dendrites. It is noteworthy that following complex spike activity, the simple spikes may be subdued by the potent complex spike input. Moreover, Purkinje cells showcase spontaneous electrophysiological activity characterized by trains of spikes dependent on both sodium and calcium. This fundamental behavior was first elucidated by Rodolfo Linas, serving as a cornerstone in understanding the intricate functionality of Purkinje cells. Remarkably, P-type calcium channels bear the moniker of Purkinje cells, underscoring their pivotal role in cerebellar function. Their activation by climbing fibers can transition Purkinje cell activity from a quiescent state to a state of spontaneous activity, acting akin to a toggle switch. While studies have contended the toggling phenomenon induced by climbing fiber inputs mainly occurs in anesthetized animals, recent observations have indicated the presence of Purkinje cell toggling in awake felines. Notably, a computational model of Purkinje cells has delineated that intracellular calcium computations underpin the toggling mechanism observed in these neurons. Furthermore, research findings have proposed that Purkinje cell dendrites release endocannabinoids which transiently downregulate both excitatory and inhibitory synapses, shaping the overall synaptic activity. The intrinsic mode of activity in Purkinje cells is predominantly governed by the sodium-potassium pump, suggesting a more intricate role beyond maintaining ionic gradients. Notably, perturbations in this pump, such as mutations leading to rapid onset dystonia Parkinsonism, underscore the criticality of this element in cerebellar computation and overall brain function. Additionally, experimental observations using uabine to block Na+, K+ pumps in live mice have induced symptoms of ataxia and dystonia, indicating the profound impact of pump dysfunction on motor control. Mathematical modeling of experimental data has suggested that in vivo, the Na+, K+ pump generates extended periods of quiescence in Purkinje neuron firing, with potential computational significance. Disturbances to Purkinje cells in humans can stem from various factors, including autoimmune diseases or genetic mutations leading to conditions such as spinocerebellar ataxias, gluten ataxia, or Unverricht-Lundborg disease. Furthermore, neurodegenerative disorders like multiple system atrophy impacting the cerebellum can also precipitate the degeneration of Purkinje cells. Diseases like gluten ataxia underscore the irreversible damage caused to Purkinje cells by gluten exposure, emphasizing the necessity of early diagnosis and adherence to a gluten-free diet to ameliorate symptoms and slow down disease progression.